3. Molecular diagnosis, right? What it is? Molecular diagnosis. Now listen this statement. To treat a disease effectively. What? To treat a disease effectively, early diagnosis should be done. What should be done? Early diagnosis should be done. Is it okay? Number 2, pathophysiology should be known. Pathophysiology should be known. Therefore, to treat a disease effectively, number 1, what required, sir? Early diagnosis. Early diagnosis. Number 2, pathophysiology. Pathophysiology should be known. Is it okay? Therefore, early diagnosis should be done. Pathophysiology should be known. Is it okay? Then only we can treat a disease effectively. But you take conventional diagnostic methods by conventional methods of diagnosis. Conventional methods of diagnosis. Sir, what are conventional methods of diagnosis, sir? Serum analysis and urine analysis. What and what, sir? Serum analysis and urine analysis. They comes under conventional methods of diagnosis. By conventional methods of diagnosis, right, early diagnosis is not possible. What is not possible? Early diagnosis. Possible, not possible? Not possible. Early diagnosis. Early diagnosis not possible. Early diagnosis is not possible. Sir, these conventional methods of diagnosis right, they identifies the pathogen only when the pathogen shows the disease symptoms. Therefore, pathogen identified. What, sir? Pathogen identified only when pathogen produces what, sir? Disease symptoms. Therefore, by conventional methods of diagnosis, a pathogen is identified only when the pathogen produces the disease symptoms. But by the time pathogen produces the disease symptoms, right, the number of pathogens in the body, right, in the human body will be more, right? Therefore, by this time, sir, what is the meaning of by this time here? By this time means by the time pathogen produces the disease symptoms. Okay. By this time, number of pathogens will be more. Number of pathogens will be more. Is this statement okay? Therefore, conventional methods of diagnosis, what and what? Serum analysis and urine analysis. By these conventional methods of diagnosis, early diagnosis is not possible. pathogens are identified only when the pathogen causes the disease symptoms. But by the time pathogen produces the disease symptoms, then the number of pathogens will be more in the human body. Is this statement okay? Early diagnosis is possible by recombinant DNA technology. Recombinant DNA technology, PCR, polymerase chain reaction, ELISA, enzyme linked immunosorbent assay. These are used for early diagnosis. Used for early diagnosis. Early diagnosis possible. Early diagnosis possible by what, sir? Recombinant DNA technology. Recombinant DNA technology, that is rDNA technology, genetic engineering. Number 2, PCR, that is polymerase chain reaction. Polymerase chain reaction. Number 3, ELISA. ELISA, this is new for us. ELISA. ELISA full form I am writing. Enzyme linked immunosorbent assay. Therefore, what is the full form of ELISA, sir? Enzyme linked immunosorbent assay. Is it okay? Right? This ELISA you will get in detail in zoology. Direct ELISA, indirect ELISA you will get in zoology. Right? Now, sir, early diagnosis, right, it is possible by rDNA technology. What more, sir? PCR. And what more? ELISA. Is it okay? Right? Now, listen this statement. PCR. What is the full form of PCR, sir? Polymerase chain reaction. Polymerase chain reaction. Sir, this PCR, polymerase chain reaction, is one of the technique to identify the disease before disease symptoms are produced. Before disease symptoms are produced, pathogen is identified, I am saying. Before disease symptoms are produced means number of pathogens are more or less. It means even though the number of pathogens are less, before disease symptoms are produced, PCR is used to identify the pathogen. How, sir? By amplifying the DNA of the pathogen. Because PCR is used for amplification, yes or no? Therefore, by amplifying the DNA of the pathogen, even though pathogen number is less, even though pathogen has not produced the disease symptoms, pathogen can be identified. Is this statement okay? Right? Therefore, PCR used for identification of pathogen when even before what? Disease symptoms. Even before disease symptoms are produced. That is, even when number of pathogens, even when number of pathogens are less. Is it okay? How, sir? By amplification. By what, sir? Amplification of DNA of pathogen. Therefore, by amplifying the DNA of pathogen, even though pathogen number is less, even before the pathogen has produced the disease symptoms, pathogen can be identified by using what, sir? PCR. Is this statement okay? Right? One more statement we discussed concerned to PCR. Sir, PCR. PCR is used to identify PCR is used to identify what, sir? HIV. Used to identify HIV in suspected AIDS patients, right? This we discussed already. Number 2, PCR is used to identify mutations in what? Mutations in genes of suspected cancer patients. Suspected cancer patients. Number 3, PCR is used to identify many other genetic disorders. Many other genetic disorders. Many other genetic disorders. Is this statements okay, right? For ELISA. Right. Sir, this ELISA is based on antigen-antibody interactions. ELISA is based on what, sir? Based on antigen-antibody interactions. Is it okay? Right, this ELISA used to identify the pathogen. Identifies what? Pathogen by the presence of antigen or by the presence of antibodies that are produced against it. Is this statements okay? Right. Therefore, ELISA, it is based on antigen-antibody interactions. It is used to identify pathogen. How, sir? Based on presence of antigen or based on presence of antibodies that are produced against it. Is this statements okay? Right. This is concerned to what, sir? ELISA. This is concerned to ELISA. DNA fingerprinting. What it is? DNA fingerprinting. It is used in forensic sciences. Yes or no? Used in forensic sciences. Sir, why it is used in forensic sciences, sir? To solve parental disputes. To solve what, sir? Parental disputes. In hospitals, sometimes the children are exchanged. Yes or no? At that time, right, the children belongs to which couple? That can be identified by using what, sir? DNA fingerprinting. Therefore, DNA fingerprinting used in forensic sciences to solve parental disputes. It is also used for search of criminals. Search of what, sir? Criminals. Therefore, DNA fingerprinting used in forensic sciences to solve parental disputes and for search of what, sir? Search of criminals. Is it okay? Right? Now listen here. Biotechnology process. Biotechnology process in industry. In what, sir? Industry. Sir, biotechnological processes in industry called as white biotechnology. What it is called as, sir? White biotechnology. White biotechnology. Biotechnology process in agriculture. In agriculture called as a green biotechnology. Green biotechnology. Therefore, biotechnological processes in agriculture called as green biotechnology. Biotechnological processes in industry called as a white biotechnology. Is it okay? Right? Now go for the next topic. Biosafety. Biosafety and ethical issues of genetically modified organisms. Biosafety and ethical issues of genetically modified organisms. Right? Now listen here. Sir, genetically modified organisms, they are having number of uses despite of its several advantages. These genetically modified organisms, when introduced into the natural ecosystem, they are giving unpredictable results. What, sir? Despite of several advantages. Despite of several advantages, genetically modified organisms are having or giving unpredictable results when introduced into natural ecosystem. When introduced into natural ecosystem, it is giving unpredictable results. Sir, what are that unpredictable results given by genetically modified organisms? Number 1, transferring allergens or toxins to animals and humans as a side effect. Sir, when these genetically modified organisms are taken as food materials, by animals and human beings, that genetically modified organisms, they are causing allergy, they are becoming toxic. Is it okay? Right? Therefore, transferring allergens or toxins to what, sir? Animals and humans as a side effect. Is this statement okay? Right? Next, number 2, risk of changing fundamental nature Risk of changing fundamental nature of vegetables. Risk of changing what, sir? Fundamental nature of vegetables. Risk of changing fundamental nature of vegetables. Next, number 3. Harmful effect on biodiversity. Harmful effect on biodiversity and adverse impact on environment. These genetically modified crops, they are having harmful effect on biodiversity and what more, sir? Adverse impact on what, sir? Environment. Adverse impact on environment, right? Next, number 4. Sir, we are discussing biosafety and ethical issues of what, sir? Genetically modified crops. What they are? Genetically modified crops. Yes or no? Right? Sir, these genetically modified crops, they may change the natural evolutionary pattern. May change what? May change natural evolutionary pattern. May change the natural evolutionary pattern. Next, number 5. Sir, there is a chance of gene pollution. It means genetically modified what, sir? Organisms. They may cause what, sir? Gene pollution. They may cause what, sir? Gene pollution. Is these statements okay? Right? Thank you.